Hi, and welcome to the Virology Control Studies Project podcast. I am joined here today. It's a very special episode with Marvin Haberland. Hi, Marvin. How you doing?
Hi, thanks for inviting me, Jamie.
No, it's great to have you here. It's been a long time in the making. We've been ships in the mist, kind of missing each other, but it's great to have you on. Now, Marvin and myself have been doing some similar projects over the course of the years, tailoring control studies and working in a legal capacity. Marvin is obviously most notable for standing up against the German authorities in a masking court case that eventually got overturned. And also, his company that he runs next were involved in some of the controls that Dr. Stefan Lanka was doing in 2022-23. Could you tell us a bit about Next Level's involvement in those control studies and kind of what excited you about getting involved in that project with Stefan?
Sure. Everything started with the so-called measles process, court process in Germany around 2015-16, when Stefan basically asked in the public. He published like a winning game to win money, 100,000 euros for the proof of the measles virus. And he had some rules to get the money. So the publications should be, of course, scientific publications from the Robert Koch Institute, which in Germany is the Center for Disease Control, and has also by law, they are obliged to carry out any sort of scientific experiments to basically search and research viruses. So that's why he made this as a rule for the 100,000 euros prize money. And there was a doctor handing in some publications and asking for the money. And then, of course, Stefan basically did not pay the doctor, and he said he assessed the evidence. He said, okay, in these publications there is no evidence, so it came to the court. And for this court case, Stefan carried out also some control experiments for the basically cytopathic effect of the measles virus, and also for the genome sequencing to show that the results in the cytopathic effect and also in the genome sequencing process, which is the genome sequence of the measles virus in the end, are not specific to a ill sample, for example, from somebody who has measles condition. And he handed in these results to the court as well. And our job basically in Next Level, and before that we were our name was Corona Fakten. So we published these results after Stefan has published them. We published them as well, and we supported Stefan to spread them in various different social media channels, and wrote some publications that were easily accessible also for laymen, people that were not experts. And that's our role.
Because that's how I came across them myself, because
Yeah.
obviously Stefan on his Wissenschaft plus magazine that he runs. I mean, I think he put the controls behind a paywall there, but it's to a not. I mean, I speak a little bit of German. My mother lived in Germany for about 10 years, but not good enough to kind of read and appraise, scientific publications. So what Next Level did was, or Corona Fact, and then was, put it out in English and make it available. And that's where I came across it. And it was kind of, for me, it was like, okay, well, learning about virology as it was then back in, during the height of the scandemic, it was okay. Well, this is what needs to be done, right?
Right, right. And then another thing that we did for the measles topic is we published a big documentary film that was also published in a private cinema in Germany, but is also available on our website, showing the complete measles case with all the details, with everything you need to know about this process. Because if you research this process in the German media or internationally, you will come across various different claims that the process was only won by Stefan because there was 5 publications handed in, which together proved the measles virus, but Stefan asked only for 1.
That's
This
right.
is basically how they portray
Well,
the
it
case.
was Reuters that ran the kind of the cover story, wasn't it? To kind of baffle the. It was a very clear judgment at the higher court, because it was over 2 courts: the lower court of Ravensburg and the higher court of Stuttgart. And at the higher court, the conclusion was categoric. It was there is no evidence of the measles virus in any singular publication. I looked into that quite deeply myself, and that the part that Reuters latches onto is from Dr. Podbielski, who was the supposed expert. I mean, he was a microbiologist, really, that made a few kind of category errors in over the trial, right? But he, at the lower court, made an opinion that together these papers would constitute evidence. But because the lower court judge was somewhat partisan or biased, should we say, they didn't let Stefan have a rebuttal. So that opinion was just an opinion and wasn't actually adjudicated on. Is that correct?
Yeah, that's correct. And what is very important also for the audience to know is, of course, there is the official statement of the higher court. But what is more interesting is the original protocols from the court case, what was said during the trial. And here we can find that this Dr. Pobielski, which was the expert witness, a virology, head of virology from the University of Rostock in Germany, he said in the first trial in the lower court that there are negative controls lacking in all of the scientific studies publications.
So he
that is what
agreed
he
kind
said.
of. He's agreed kind of, and because that's been lodged into German law, as a precedent, those are quite huge words, right? Essentially admitting that the processes of isolation are fraudulent or pseudoscience. If you cannot,
right.
For me, when I've been developing the project and developing what we're doing with control studies, it always seemed to me apparent that the control is really the vehicle to operate legally, within science. So you have to have a randomized placebo-controlled trial for drugs if you're to do it in the positive, and also controls of a negative control. Certainly, I mean, positive control sometimes, but without doubt a negative control must be done legally. Would you say, would you agree with that?
Yeah, I totally agree with that. And this is also, I think it's a non-negotiable. Everyone would agree.
Yeah.
Even the virologists that we asked over the years always agree to that. There is some, debate on what is a correct negative control and what is not a correct negative control. But as far as the negative control itself, they all agree. And obviously in Germany, what is also something very good is for all these cases, also court cases, that we have the law of infectious diseases or Infection Protection Act. And it states in the first paragraph that everything should be according to the scientific method, which is pretty a strong support for our claim that that it is not scientific. Because if you then look up what the German Research Foundation says, which basically publishes the rules of science for all of Germany, for everyone who gets research funding, from tax money, and so on, like for the Robert Koch Institute, for universities, they all have to follow these guidelines. And they say also that every experiment has to be controlled, every method has to be controlled. Ideally, every experiment has to be blinded, and so on, to reduce the bias. So this is quite clear. But this guy, Dr. Pobielski, expert witness in the case, said that all of the publications handed in by the doctor to claim the 100,000 euro prize money were lacking negative controls. And on top of that, this is not written in the final statement from the court, which is a pity, because it's a very important thing that he said. And then he said another important thing, which is also not in the final report. That is that the process of genome sequencing is not a proof for a virus itself.
And I wanted to touch upon that as well. Because I went through the lower court of Ravensburg. I went through both because they published the jurisdiction of the higher court, and you can kind of read, and I translated it from jurist.de. I think all of the higher court, they have to publish kind of word for word, but the lower court, where all of these things, because really the higher court was just procedural, because it was based on the one paper claim, and they just went, well, quite clearly there is none, because at the lower court he admitted that there was no single paper which satisfied all of Koch's postulates.
Right.
It was more procedural, that they didn't really adjudicate on anything. They just went, well, this is quite clear. And so it was unanimous. But like you say, I think it wasn't really recorded, because it was only in kind of some PDF notes and things where cross-examination, Dr. Podbielski made a lot of these claims, right? So
yes.
So have they managed to kind of skirt around that by saying, well, those claims aren't actually lodged into German law, or like, do you have to kind of go into it? And also, you just touched upon a very important point, that one of those papers that was submitted by Dr. Bardens was the genome sequence, the supposed whole genome sequence of measles, and kind of maybe inadvertently a bit, maybe they didn't really realize in the higher court that by adjudicating that there is no single paper that proves the existence of measles, they are agreeing that the whole genome sequence of measles does not constitute its existence, which means any PCR test, for instance, does not prove the existence of the virus, does not prove that you have this thing.
This is written in the official protocol from the trial from the lower court. So if you want, I can send you that one as well. So it is a document that can be used in legal context in Germany. It also has a reference number. So I use this quite often on my own, this document, which is far more interesting than the final report of the higher court, because, as you said, they just followed the one publication theme, and I think they did that to hide all the information, all the explosion power that was inside the first level, the first court. So they try to make up another argument to let Stefan win, but not because of the scientific arguments, but because of something different. And very interesting also, the doctor, Dr. Bardens, who claimed the money from Stefan, he tried to object the higher court. He tried to go to the third level, to the Supreme Court of Germany, basically. And Bundesgerichtshof is the name. And this highest court did not accept the objection. So they probably saw that this would be too
damaging
for the virology sector or whatever. I mean, these are conspiracy theories, obviously, but you can only come to this conclusion if you read.
You can only agree. I mean, I've heard because it was a unanimous decision as well, because all three judges at the higher court made that unanimous decision on a procedural thing. That I mean, maybe that was the get out clause as well, to not have to deal with it. Stefan, submitted, I think it was about a 70-page, manuscript tearing apart virology, and submitted. They were kind of the rudimentary controls that led up to the controls that he did based on the SARS-CoV-2 isolation in 2022-23. These were kind of a stripped down version. Is that correct?
Yeah, that is correct.
Okay.
He during COVID he went to a public lab in Switzerland and carried out with them the control experiments again, basically for SARS-CoV-2. So initially he planned to do a three-step approach for the cytopathic effect first, then second for the genome sequencing, and third for the EM pictures. As I said, this was carried out in Switzerland, and we were not involved in the experiment themselves.
Okay.
These were carried out by the lab, public lab there. But we were again.
So it was commissioned by Stefan. It wasn't Stefan himself that carried them out. Yeah.
Yeah, exactly. And what our mission was, again, we supported Stefan in publishing these results, even in our magazine. The first magazine from Next Level 2023 is also available in English. We published the results from the cytopathic effect. But for the second and third step, the results have not been published so far. This is
because I was going to ask you about that, because that's really the journey that I've been on, because I saw your wonderful publication back in early 2023 when the controls were released, and I read the first part, which was fantastic. It was like, right, so there is no benchmark. I mean, really, when you kind of analyze it, after we've completed, the first two parts as part of the control studies of doing the electron microscopy, really there is no much more biology left. Once you kind of leave the physical particle behind, you're then into a completely another realm of science, which is genetics and chemistry. Really, it has nothing to do with the actual biological particle itself. So
if there is a particle, this is very important, because it could be artifact, or it could be some other structure. We don't even know if these are particles. They could, but it's difficult to prove.
This for me was kind of my journey, is that I found this cell culture work, and it was mind-blowing to me that's what needed to be done. And then coming on to part two, obviously there was that was missing, and then part three was a mathematician's model by a mathematician in Hamburg, who theoretically put together that if you did a reverse sequence of essentially any genetic sequence and lowered the read length and lowered the Kmer value for alignment, that you could essentially create any genome mathematically.
That's correct. Yeah.
Now, obviously, the rebuttal to that amongst geneticists is, well, we don't lower the read length. They do it at about 150, and this mathematician was kind of lowering it to somewhere in the region of 30 or 40 base pairs, which wouldn't be acceptable in the mainstream. But still, if you can show that even lowering the accuracy levels, you can essentially create anything. You shouldn't have a science which is predicated on having to do it at an exact level. It should be either you're reading these genomes left to right and you have a whole genome, or not. Like, I don't think that, but really, to me, the genetic sequencing part to it was the end goal, and so that's been kind of my mission for recreating that. So Stefan did do that. He did do the genetic sequencing, but didn't release it to the public. Is that correct?
Yeah, that is correct. So he has not published it so far, but I can say that he said that the results show that there is a 98% accuracy, or like to
Yeah.
the genome
Yeah.
overlap, without using any supportive methods like PCR primering or other methods. Just
Interesting.
by the supernatant genome sequencing itself,
Interesting.
already 98% of the genome is covered. This is what I can say, but yeah, it has not been published so far. I don't know if or when it will be published. I cannot say that. I don't have information from Stefan regarding this, but the other thing that we carried out and published is what you just said, is the genome sequencing of the raw data that was published from Wuhan, China, from the publication from Fan Wu et al. From the SARS-CoV-2 genome reference genome, and we showed with the mathematician, we published this, and we showed, as you said, that there was it was possible to show the sequences from multiple sequences from different viruses
from
these raw data.
The raw data of the initial.
Exactly, yeah. And you said yes, but the KMAR value, the software parameters in the assembler, or in the assembly process, of course they are. You can argue that they are kind of subjective because they are not always using the same values. Also, the values are not, usually they are not published. They are not part of the publication. They are part of the raw data, which you have to ask the authors for. The KMAR value is usually never published.
Yes, that's
and
interesting.
the correct software, the software parameters used are not published. This is one problem. The second problem is the bioinformatics that do these kind of assemblies and alignment processes, they are never blinded, really. So this is a problem in itself. Then they only have one culture or one data set, because they don't have a negative control, a correct one. They only sequence the so-called infected culture. They never sequence the negative control, which doesn't.
So using all of the reagents and all of the parameters to conduct a side-by-side comparison.
Right. And then there is another thing. They can argue, yes, we use another base length or another KMAR value, but they use very dirty techniques. They run multiple cycles of PCR with different primers, and they create they create a sample which is much more likely to provide the results.
They used the thing called the Takara protocol for the original SARS-CoV-2 sequence, and it contains huge libraries of primer sequences and huge parts called a library prep, which is essentially, they get things like there's a part called a poly A tail, which is like A in it, and they know that supposedly these RNA sequences contain these set kind of codes, and they have a poly T adapter cell. So they're designing these primers and these adapters for what they're looking for, and so these huge kind of sets are very circular reasoned that, they are very specific and very bespoke things. And then all of a sudden they're finding the things that they're looking for. And so this isn't really science, is it?
Right. And another circular reasoning is the alignment process itself, because in the alignment, usually you use the template from another reference genome, and this is of course also circular reasoning, because you are aligning your data set, and basically you tell the software how to align it, what you are looking for, and you could use any other template, of course.
Yeah, you're trying to kind of force it into that mold that already exists, right?
Right, and then you fill up the gaps with some other
with the junk that's kind of in and around.
So
I mean, there came a value in itself, the De Bruijn graph method of alignment, which is where you take small chunks and you slightly alter the beginning and the end of each one.
right.
Means in and of itself that you are not sequencing anything in reality,
Exactly.
because if you were to take one section, you would expect the second section to fit, but they're not. They're sequencing it and moving them around so that they are intentionally putting different organisms together, and then just saying that they should be indicative of something that's in there, right?
Yeah. All in of itself, even if the process was scientifically sound and carried out correctly, it can never be proof of an existing entity, because if at best it's an indirect proof, like a
Yeah,
surrogate marker.
exactly.
It can never be proof of an existing entity. The only way you can basically design genome sequence processes to be very, very specific is that you have the particle, the real existing particle, you already have it isolated, and you show that the sequencing is always the same result, always specific and independent of every other variable.
Yes.
And this has never been done because they don't have the isolates, they don't
No.
have the real particles. So it's all based on a modeling approach and a statistical game. It's not a proof of an existing entity. And this is also what, as I said in the measles court case, the expert witness admitted that it is not a proof. And for SARS-CoV-2, our Center for Disease Control in Germany, Robert Koch Institute, in their internal protocols that were published after some time, they said this as well. They repeated this inside the protocols. You can read that the German Ministry of Health and the Robert Koch Institute say that genome sequencing is not basically virus proof.
Yes. Yeah.
This is not the definition of it.
That's massive, really. And has that helped you in your own trials?
Yes.
You're
Yeah.
saying that
I always
you're working recently. Obviously, you came to notoriety with the masking case, where you were going around and tapping policemen on the shoulder saying, look, I've got no mask, can you please please book me, and taking all of these fines, and took a whole binder of information, and they got scared. They got scared, Marvin, that you knew what you were talking about and had to drop the case. Are these the types of things that you were trying to submit into court?
Yes, for the mask mandates, I used several different evidence proofs. I mean, official publications. I used the court protocols from the measles court case, and various different arguments. I think the most important argument is that by law, in paragraph 1 of the German law, it says that you have to work scientifically. German Research Foundation defines what means working scientifically. They say you have to control the methods, you have to be blinded, and so on. You have to publish all the documentation required to to replicate the result, and so on. So all of these things have not been done in the SARS-CoV-2 publications, and so this is already enough, okay, because it violates the law.
Yes.
For the now, we are supporting many different families and parents in Germany, because since 2020 there is an indirect vaccine mandate for measles. So you basically cannot bring your children to the school or to the kindergarten without this vaccination.
Yeah, it's awful. It's the same here in France, actually. They just kind of plowed on ahead, because that's why Stefan put out the wager in the first place, right? To raise
Exactly.
awareness of the fact that the German system were trying to somewhat coerce parents to do this to their children. So
That's correct. That was the initial motivation back in 2000. I mean, even before 2015, there were already talks in the political spectrum to publish or to invoke such a mandate. And Stefan was contacted by high level politicians. Okay. To maybe come up with an idea, maybe with then he finally came up with this prize money thing, to come up with an idea on how to bring this in the court. And this stopped it for a long time. We don't know exactly, but probably this was one of the reasons why it only came then in 2020. And since then, it's it's active. It's still a law in Germany, but of course there are many parents that do not vaccinate the children, and we support them in the court. We have around 15 to 20 cases running right now. Some of them are even in the higher court already, because the first court usually do not assess the scientific evidence, and the second level, there we try to force the judge to look into the scientific evidence. But yeah, we will see how this is turning.
So the authorities are trying to clamp down on it. Are you seeing it? Are the people that are getting into court, are you seeing them from certain kind of regions? Some regions are more hot on it than others. Are you able to advise people to be able to exercise their rights up front? Are you finding that if you know how to back yourself legally right at the very start, is that kind of something that you do to consult to people also to be able to not have to vaccinate their children?
For sure. We offer consultation in this area, and we try to support wherever we can, not only with the court cases, but also before that, with the communication with the health authorities and with the schools and so on. So
a bit like medicine, it's got to be preventative, right? Preventative before.
Usually, it always ends in the court because the health authorities do not accept the argumentation.
Yeah,
and then
it's the same here in France. This was one of the things that kind of inspired me to do what I'm doing, is because obviously I'm from the UK, and all of my family are back in the UK, and they're not quite as overt in Central Europe as kind of how much they were willing to kind of push it, because here, the same as Germany, we had a vaccine passport for nearly a year. We were banned and ostracized from society. We weren't allowed to go to pubs or restaurants or sporting arenas or cinemas, or practically everywhere, even soft play centers for my children. And so it really was quite bad. And so I empathize with my German counterparts, that it's they're really quite bad in Central Europe in terms of pushing these political atrocities, really. And so you're doing a great job. Thank you, for backing and helping these people out. In terms of just kind of coming back to the scientific part of it, we were talking about genetic sequencing in the latter kind of stages of Stefan's public work. He started to question genetics in and of itself. Now, as I've kind of developed and had a look at the results that we've been getting as part of the project, a lot of the same inconsistencies within the realms of genetics have started to appear within virology. Have you got the same kind of idea, or have you got the same skepticism about genetics and DNA and RNA itself?
Yes, that is correct. Because if you observe the scientific literature, you can see that the same mistakes are done as in virology. So usually there is no negative control, or there is no control for every step of the methodology, and this is a problem. If you carry out controls, then you basically falsify the methodology of the
Yeah,
gene. And this is what we have been publishing a lot during the past months and years in Next Level. So we found several different publications that carry out control experiments. For
interesting.
instance, in the DNA
Mm.
test of witnesses in the court, or also for parent analysis, DNA analysis,
Yeah,
and on top of that, I mean, these publications are published, they're peer reviewed, and they
mm.
show that the method is not, specific. That
Mm.
it is. For instance, there was one publication where 17 different DNA analysis from the US, all from authorized labs, they were presented with a data set from an official original court case, and all of them came out with a different result
Results. Yeah.
than the court. On top of that, where their opinions were vastly different,
Mm. I
this.
think I've heard of it. It's a group called the Innocence Project, run by a couple of lawyers, Greg Hampkin and Barry Scheck, and yeah, they did some really eye-opening work to me as well. That I started to kind of come across this because they were seasoned lawyers. I think Barry Scheck was part of O.J. Simpson's dream team on the kind of forensics periphery, and he just started to have a look at these things and just actually try and find a benchmark study or a blinded study of these. I mean, it's a huge can of worms, right? When you look at it in the legal system, people that are convicted purely based on DNA evidence, and people, the general population believe that it's 99.99999% accurate, right? And so when he went and actually challenged the three-letter agency, which is NIST in the US, and said, "Have you actually got a blinded study?" And they said, "No." It's kind of eye-opening that they can't do these studies where I think Greg Hampkin even said that it's well known that they don't like doing the forensic analysis blinded. They want to know everything about the trial.
All right.
Now, if you were a scientist, that's logically absurd, right? You would want to be blinded so that you're not biased to the outcome, right? But they want to know, who's the most obvious candidate in this? To make sure that they get the right result. And it really becomes quite spurious when you start to kind of scratch away at the surface. And
Another thing I can add to that, we carried out some control experiments in
fantastic.
Germany,
Wow.
in the area of genetics. So we sent different samples of sheep and other animals and dogs for overall to a laboratory, and blinded. We sent multiple samples from dogs, and there was also a human sample in between, and they were not even able to distinguish them, and
Wow.
all the racial tests were, or most of them were wrong. So they were not able to clearly find the race of the dog.
Okay.
And they even, with the human sample, they also, found a race, dog race. And then the same with the sheep. So this also, and this we published on our page as well. So
Interesting.
it
Interesting. I
shows
haven't seen
that.
that. I'd love to see that because I've only seen anecdotes. So I'd love to actually see that being carried out. Yeah.
Yeah, sure. And then there is another publication from a German university that we published on our channel some days ago, showing that what was done. There were around 340 data sets from different men compared with 330 different data sets from children.
Right.
And they did like a cross analysis of all these combinations over 100 16,000 combinations in the software program,
Mm.
and they found out that over 90% of the 96% actually of the children had multiple fathers according to the results, and many, or the majority had like 10 to 15 fathers. Some had even above 20 fathers from this data set. So and this was carried out in the blinded fashion because it's
Yeah.
purely in the software program.
Yeah.
And you can see that some foreign men, not related to the children at all, had higher probabilities than
Right.
the actual father.
Right. Interesting.
And the result of the study, the conclusion of the authors is that 99.99% accuracy doesn't mean anything. Okay,
No.
this is what you said before, and
Yeah.
they clearly say that in the publication.
Interesting.
Yeah.
Yeah, it's the same. As soon as you put under scrutiny using the idea of blinded or control studies, all of these sciences seem to just capitulate. It's like you say, one of the ones, there was a lady called Karen Keegan who had to get an organ transplant, and to see if they could have a donor from from one of her children, they conducted sequencing and found that her kids were not her kids.
Yeah.
And all of a sudden they have to kind of go, oh, okay, right, well, that's because, and they just come up with this thing, chimerism. Apparently she is genetically chimeric, which means that she has two sets of genes or something. They just make it up off the top of their head. Okay, well, we've got to the end of that. And one of the things that I'm starting to it's kind of a side project. I'm starting to work with HIV positive individuals, because for me HIV is a really interesting one, because they say that once you have HIV, it incorporates into your genetics. So because it's a retrovirus, apparently you have it for the rest of your life. And so really the people that are HIV positive are walking, talking control experiments, because if you ever test negative at any point in your life after being positive, it kind of debunks the entire viral story of incorporating into your genetics. And again, they found people that have exactly done that. They kind of get away with it because they have this, they apply them with antiretroviral medication as soon as they get a positive and scare you into taking it, that if you don't, then you're going to die. And there have been hundreds and hundreds of cases of people either not taking it to start off with, or at some point in their life stopping and then testing negative afterwards. And they're called elite controllers, apparently. It's a really nice term where we don't really know why it's doing something, but our scientific results don't match up with our story. So they have to kind of get away with it. And I'm working with lots of HIV positive individuals, and we're kind of, doing some of these control studies and operating a support network for people that are learning to or trying to give up these medications that they're applying people with for a fictitious virus. So
yeah, that's very important work.
Thank you. No, it's rewarding as well, because
you would think that HIV is not that it is already something from the past. It came out in the 80s and so on, but it's still running today, and they are making a good money out of it. Yeah.
Well, certainly, the market cap for antiretrovirals, because they're taken on the daily. You can't get them on insurance, and so they are making mega bucks off of these people. And the mostly homosexual community, who's most prevalent for being diagnosed with HIV, they're a heavily maligned group of people, and ostracized, and just heavily medicated. The way that the medical industry treats these people with drugs such as AZT and nevirapine, which have, on the front of AZT, it has a skull and crossbones, do not ingest. If it's dropped into the environment, it will kill all the fish.
Yeah, it's very toxic. Yeah,
it's very toxic, and so it's it also comes with the common connotation that, you know, drugs are bad, right? Because there is no vaccine. It's kind of a little bit easier to get people to understand, because these drugs are, you know, are highly toxic. They tell you, they have to tell you on the front of these things. And so, you know, that's what we're trying to do at the moment is, well, that's what we are doing at the moment is operating as a support group. And I wanted to talk to you about, you know, the way that I've kind of, I ran the project, and we open sourced it, and we crowdsourced the money to do the experiments up front, and we took video of the lab and released as much kind of information on the open source marketplace as possible. The thing that I want to kind of infuse is a new paradigm of kind of looking at science for people themselves, to develop these groups of people that come together if you are interested in these areas. Is that something that you're interested in, to try and move forward away from that kind of old paradigm that the medical institutions and political institutions have, you know, over the peer review process, for instance, over you know, conducting of science and the way that it's funded?
Yeah, certainly, because I don't think that the peer review process is bad in and of itself, but internationally it is not taken very seriously, because they are not really looking for the correct scientific methodology. They just review and pass publications without negative controls and with many methodological problems, and this is why we cannot really you know, trust them anymore. And yeah, I think crowdfunding or like a more like a public approach is a very good thing, or doing it very transparent. You know,
Exactly.
what we have been doing in the past in our work is basically assessing publications transparently, just showing, okay, why is this publication good, why is this publication bad, these are the reasons, these are the arguments. And what you are talking about is very important for somebody, for like an entity or group which carries out experiments themselves. What we only have been doing in Next Level for some genetic topics, but we are mostly assessing publications.
I get you.
So
Yeah.
this is something very important for you, for your work, because you are carrying out the experiments yourself. So there, it's very important to do or to have this environment, I think.
Yeah. No, I understand what you're saying in terms of the peer review process. I mean, I've looked into the peer review process, and I work with actually a lady who has she was tenured at Nature for 17 years, and she got so disillusioned with the you know, the process that she had to leave, and you know, the amount of kind of nepotism, you know, where it's just kind of, oh, I know you and you know me, and so we'll all kind of review each other's papers, and great. Do you know what I mean? It's kind of like back rubbing to get these things through. And, you know, the fraud is so entrenchant in that area. You see people, there are things called paper farms, right, where people lease out their credentials so that essentially you can get a paper published for money, right? So you pay somebody, and I think there was the name of a chap who's currently under scrutiny, who has been releasing, you know, publishing 5 papers a day. You know, you've got to kind of go, right, well, you know, you're either very quick at typing, or there's something, you know, there's something not right here.
Like the peer review of Christian Drosten's PCR test of COVID took like less than 48 hours.
48 hours, yeah. And it's rubber stamped, and it's then kind of moved on. And, you know, I think we've got to challenge this area, because it's a methodological you know, breakdown. It's a breakdown in the system and the structure. And like you say, intrinsically, there's nothing wrong with the scientific method. Intrinsically, there's nothing wrong along with the peer review process. It's just become corrupted to the core, where we really have to start to kind of unpick it at its root.
Right. And another thing I want to add quickly is that what we did with Stefan with the control experiments in SARS-CoV-2 is not only to publish them and to to do these kind of things, but also we basically performed all the communication with the virologists in Switzerland, for example, and in other countries, to before these experiments were carried out, we contacted many virologists and asked for a cooperation to carry out those controls together with them on our cost, and film it, document it, and all of the virologists from all the high level institutions all disagreed. Nobody was
that's what's so interesting. We've released the results, and it's been coming up about a year, and I've been through exactly the same thing. Please, I would like a rebuttal. Please find the methodological flaws in our experiments that we've run, and it's just tumbleweeds. And yes, like you, it's come to the same kind of crossroads, where I've been speaking to tenured virologists and said, I need to conduct some more controls, and I'm more than happy for if I pay you, for you to be happy with the process. Because we even took video. We took video of the of every single methodological procedure. We logged every single transit, dry ice, transit between the laboratory that conducted the cell cultures in transit to the TEM. We logged every single bit, far more than any peer review paper would ever do, right?
Yeah, you did a very great job.
Thank you.
I reviewed many of your work, and it's I think it's first time ever seen such a high level of documentation.
Thank you. No, that means a lot. Thank you. Thank you. And yeah, like yourself, I got to the point where I said, I will pay you, because we would like to continue doing some more of these studies, and they just go blank. I mean, obviously, they quite clearly know, right? They quite clearly know that if they can start to do any of these legitimate controls, because coming right back to what we were talking about is they claim to kind of do controls, but they call them mock controls. This is always the way that they skirt around the subject, because they go, oh no, we've conducted controls. Yeah. And you look in the method section, and it's a mock control, and a mock control is just kind of double speak for something where the parameters aren't quite the same. You find within the cell culture work that more often than not they will remove the fetal bovine serum, which we found is the variable that causes the cytopathic effects. So they're running it side by side, and they're reducing the serum in both. Well, that's not really a control, is it? That's just, I don't really know what it is, but that's the way they kind of skirt around these things. And
another problem, if I may add, is that in some mock controls, what they do not do is they don't add sample to the cell culture. So the other cell culture, the experiment, they add a sample, which of course aids the cytopathic effect, as you can see in Stefan's control also with the yeast RNA. By adding just the yeast RNA, he was able to create an enormous cytopathic effect, And just by a mock control adding nothing to the cell culture is already is not a negative control, because you have to add at least a sample from a healthy subject,
exactly,
without symptoms or something.
Yeah, because the way that the cell lines grow is very kind of in a symbiotic manner. When you grow them out onto a petri dish, they form a very kind of tight-knit pattern. You can't overgrow them where they crowd each other out, because they start to die. And if it's too dispersed, then they need some sort of kind of coherence.
A balance.
they call it confluence, right? And so when you're adding, and we did do this as part of our control experiments, we added healthy sputum just to show the difference. Is that when you add a different cell line, it starts to muck around with the way that the cells themselves just interact. It's got really nothing to do with pathogenicity. It's just the way that they kind of form a monolayer on the petri dish. And so introducing those variables obviously is a must if you're going to control them. So yeah, great point.
And what is also important to say is, you can carry out all these controls, you can do them, but in the end, it's still in vitro. So what is going on in the petri dish is not a representation of the real animal or real human. You can assume that it is, but it's not proof by itself. So this is also important. You can have the perfect control experiment, but again, it's not in vivo.
No, that's right. So, completely. I mean, so some of the criticisms that I get leveled at our experiments is, the results of them, aren't legitimate because it's kind of a pseudoscience. But I agree. We're not really showing that the cell culture work is legitimate for anything. We're falsifying it, and I think that that's the power of falsification as well.
Exactly.
Is that we are just saying this does not work. It's not proving it, and really, I think it's deeper than that as well. Is that I think that if the halls of science were to focus more on falsification rather than always trying to prove these positive claims, I think that science is a lot more kind of powerful in that realm of falsification. And so you're just saying this is certainly not, because there can be certainty to it as well. This doesn't affect this, and there's a kind of concrete evidence that you can mount off of. So that's our thinking anyway.
Basically, in my scientific training at the university, one of the first things I learned is that science is all about falsification. Falsification. This is your first goal, to falsify.
Exactly.
Okay? Yeah. So
great. Well, thank you so much, Marvin, for your time. It's been wonderful meeting you, and good luck with all of the work that you're doing in court and helping people. It's a fantastic job and a fantastic service that you're providing people. Parents being able to back themselves to not have to inject their children with horrible toxic stuff. The lives that you're saving, the damage that you're undoing is incredible. Thank you. I'll put in the show notes links of how people can find you. Where are you active most? Are you on social media or able to be contacted in any of these places?
Yes, we have our Telegram channel, which is our main channel, and then we have a website. Basically, we have published a new app called Nexica, which represents our work. It's kind of a chatbot like ChatGPT. You can communicate with Nexica, and it is connected to our database containing all of the information of Next Level, and you can basically find out about virology, other health topics. It's very interesting. So it's also available in English, so it can be interesting.
Oh, fantastic
for your audience.
Fantastic. Thank you. I'll go and check that out myself. It seems like a wonderful tool. Thank you again, Marvin. Thank you for your time, and hope to speak to you soon. Stay in touch.
Thank you too, Jamie.
Okay. Bye-bye.
Bye.